Let's take a look at the important terms page for the muscle physiology and reflexes lab. We're going to start with the muscle twitch. Muscle twitch is a single isolated above threshold stimulation of a muscle fiber that's then followed by a contraction. You will find if you stimulate a muscle that a very small stimulation is usually not followed by any kind of response. You must hit a threshold amount of stimulation in order to get a response. And then depending on how long the stimulation lasts, you will get a certain duration for your muscle contraction. What you should have recorded in the lab, and that to me is the most important part of the lab, is a muscle twitch. And the way we do this in the lab is we have skin electrodes that are being attached, and then you're sending a small electropulse to the muscle, which is then answered by the muscle with a muscle twitch, and that you can record. And what you should record looks something like this. Let me see if I can draw this. So we're going to have a split window basically. So I'm going to do two windows. It's not too easy with this fat pen here. But anyway, so down here, we're just going to send an electropulse. And I'm just going to make it like this. So this would be an electropulse. And that electropulse is followed on the muscle side. So here we're going to have the tension. The muscle is at first at rest, it's at rest, at rest, at rest, and it gets stimulated and goes boom up and then comes down. That's what it looks like. So what you will see is that at the point when the muscle is being stimulated, nothing happens at first. So if you go up here, you'll see that there's still a short lag phase, and that's exactly what this phase is called, lag phase. So I'm going to make an arrow here, lag phase. And once we start the contraction, so from here to here, just make sure that you understand that this is going down to this area. This here would be the contraction all the way up to the peak. So the muscle is developing tension, it's contraction. And once we hit the peak all the way down to the baseline, that would be relaxation. The approximate timing of this is the lag phase should be about 10 to 20 milliseconds long. So I'm just going to put about 20 milliseconds. Not sure if you can read this, but the contraction phase should be somewhere around 60 to 80 milliseconds, depending on how fresh your muscle is, or whether it's fatigued or not. So milliseconds. And the relaxation phase should be about, so on the order of 100 milliseconds. If you're recording it with a surface electrode. So total timing from start to finish would be about 200 milliseconds, something like that. Okay, so that will be the main goal of the lab, to record something like that. There are a couple of videos posted in your Canvas folder for this lab. Please take a look at it. There's one recording where it's done with a simulation, the PhysioX, which you can do on your own too. You can just type in Google PhysioX, and there should be some free versions out there where you can try that out. It looks just like in that, I think it's the second video that's posted. The first one is the gastrocnemius muscle, where somebody is using a frog muscle and using the gastrocnemius muscle from the frog to get the muscle twitch recordings. So you should take a look at both of those recordings. They're not too long. I think 5 minutes each, something like that. But that shows you exactly what we would have done in a lab if we could be going into a lab doing the actual lab work. Now, once you're done recording a muscle twitch, I always encourage students to also try sort of multiple stimulation. What happens first, I'm going to just have one, let me go down here. Let's start fresh here. So if you have if you're stimulating very quickly together, so then you will find that the muscle, let's just do a recording window like this. What you'll find if the stimulation is very close together, that the muscle starts to develop tension and it wants to relax and it can't. You don't allow it. And you stimulate again, it goes a little higher, and then it goes even higher. So there's sort of the staircase effect, and that's just what it's called, the staircase effect. If you get the stimulation even closer together, then you will find that the muscle starts to kind of weave around like this. So that would be an incomplete tetanus, where you're still trying, the muscle's still trying to relax a little bit. And then a complete tetanus would be when you don't see the squiggly line anymore. It's just a smooth, sustained contraction. And the muscle can hold it for a certain amount of time, but then it will start to run out of ATP and fatigue will set in. And that is seen as a drop in tension like this. So let's match these terms here. Wave summation, that's this part right here. Incomplete tetanus will be up here. And complete tetanus is going to be up here. And then fatigue is when the muscle starts to lose tension because it's running out of ATP and also typically building up lactic acid. Okay, so that would be all the main parts of the lab. Then there's also, we can do some electromyography. That means that you are recording the electrical activity that's going on in the muscle. That is actually done sometimes to study whether somebody has some skeletal muscle issues and or some nerve damage maybe that led to muscle weakness. So electromyography is done as a diagnostic tool, a diagnostic measure, and there's another video in your folder for this lab that you should take a look at, where a doctor is performing electromyography on a patient and is explaining how that works and what kind of information you can get out of that. Now, dynamometry means that you're recording the strength of a muscle. So that would be like the clench force that you can generate with your fist, holding something, holding an object, and that can be recorded as well. A motor unit is a complete unit of a motor neuron plus all of the muscle fibers that it innervates. So motor units, they can be fairly small, like in your fingertips where you have a lot of fine motor skills, or they can be pretty large, like in your thigh muscles, for example. So there are different sizes of motor units, but a motor unit per se is a motor neuron plus all of the muscle fibers that it innervates or can stimulate. Motor unit recruitment is how many of these motor units you're using for a certain movement or for certain mechanical work. Mechanical work, I think everybody's clear about that, is you're just moving something, lifting something, or doing some sort of mechanical work. Now, let's move into reflexes. So a reflex is defined by there are neurons involved that don't use ascending or descending tracks. So a reflex is that you're involving a sensory receptor that transports information toward the central nervous system, but then instead of using ascending and descending tracks to make an evaluation in your brain and then make a response, formulate a response, you're staying instead at the level of the spinal cord usually and sending the motor output straight out. So a basic reflex, like the knee-jerk reflex, would look something like this. You would have a sensory, let's make a patella bone and the tendon right here. So you're tapping, I can't even draw this with this fat pen here, but anyway, so you have your reflex hammer here and you are tapping the patellar tendon, let's say. That's a sensory input that then via sensory neuron, is one of those that have the cell body in the middle, gets sent to the spinal cord, and then you're entering the spinal cord. Here, let's make the spinal cord. So in the posterior horn, you're entering here. So let's draw this. Not very pretty, but it will work. So you're entering the information will go from the sensory input via sensory neuron into the spinal cord posterior horn. It will synapse typically with an interneuron. So here you have your sensory neuron, this is the interneuron, and then here you have a motor neuron, a multipolar motor neuron, and then it goes back to the leg, and then your leg, you will have this knee-jerk reflex. The beauty of these reflexes is that it's just really fast. And so you will involve an afferent neuron. That would be the sensory neuron that's transporting the information toward the central nervous system, the spinal cord in this case. And then an efferent neuron, that's the motor neuron that brings the information away from the central nervous system back to muscle tissue, and then the muscle will contract. Some of these reflexes, they can be ipsilateral, that would mean on the same side of your body, or contralateral on the opposite side, all of that. You can take a look at the videos that I posted for the reflexes. There is a short but very informative video posted about reflex testing. You should take a look at it and see if you can get some of these reflexes on yourself or on a partner. It's easier usually to work with a partner, but we're all supposed to isolate right now, so try on your own if you're living by yourself. The knee-jerk reflex, you should definitely be able to get that, and several of the other reflexes you should also be able to see if you can get those. So be sure to watch that video, especially the one on the reflexes. I really like that one, where there's a doctor performing a whole set of reflexes. Now finally, a muscle spindle, sort of that's the idea about that is that muscle spindles, they are basically skeletal muscle sensory receptors within the main part of the muscle that are primarily there to detect changes in the length of the muscle, and they help you with your fine motor control. Now, intra versus extrafusal fibers. Let's start with the extrafusal fibers. Extrafusal muscle fibers, they are basically the bulk of the muscle, and they are the ones that are generating force in the muscle when the muscle contracts. The intrafusal muscle fibers are sort of deep in the muscle tissue, and they have stretch receptors that that basically transport more sensory information. So here is what I would suggest how you should go about this lab, doing it at home. You should definitely look through the two videos that I posted about the muscle twitch. One is a PhysioX recording, the other one is a gastrocnemius muscle from a frog, the recording. Then there's also a video on electromyography and how that's done on a patient. And finally, there is another video that is explaining, it's about reflexes. And that one, I really like that one, especially because you should be able to do some of these reflexes on yourself and see how that works. Then there's also a posting of a new Labster virtual lab that is on skeletal muscle and how it works. Please be sure to take a look at that one too. That should help you to understand the material. Now, if you don't get to that one this week, that's not a big deal, but you should start getting used to the Labster format of the virtual lab. It takes a little while to get used to it, but once you get the hang of it, it's actually a lot of fun. At least I had fun with it, trying it out. And be sure that you do the Labster exercise on the action potential. That's really important, and I put that more together with the lecture content, because the lecture was on the introduction to the nervous system, mainly the action potential, and I really like that Labster exercise about the action potential. It really helps you to understand how the ions are, which way the ions are going, and how that affects the shape of the action potential. So please be sure to take a look at those Labster exercises, especially this week, the one on the action potential, and have fun with the reflexes.